Thank you, Dr. Rubin. We'll ask Dr. Fagan, Dr. Ricci to come up, and what we'll do is we'll take questions from the audience, and we have questions that have been sent in. So remember, gastro.cnf.io to send up. And I'll post some questions that have been asked.
Can I just clarify?
Surely.
So clearly, immunogenicity is a big concern for my fellow panelists, and it's something that's in the statute, that the data to support immunogenicity of the biosimilar has been thoroughly evaluated. For the infliximab and the adalimumab products that have been approved as biosimilars, as part of the clinical study that was conducted in the RA population, they did receive what's called a single transition. We don't like to say switch because that we don't want to confuse interchangeability concept, but so the patients that did receive the reference product were switched to the biosimilar, transitioned to the biosimilar, so they received it. So this was evaluated, and the immunogenicity in those patients was looked at, the PK. And so interchangeability is, if you look at the FDA guidance that was recently published in January, it describes the minimum requirements for switching that would be necessary in a clinical study to support interchangeability. And there has to be at least 2 switches back and forth from the reference product to the biosimilar. So these are different concepts. And so just so everyone in the audience realizes, the data packages that supported the approval of the 2 biosimilars did include an evaluation of the switch from the reference product to the biosimilar.
Transition.
Brian, you have a comment?
Yeah, I just wanted to talk about the issue of immunogenicity, and the devil is in the details as far as the assay goes, because any good chemist can measure drug, and you can either take patient samples and spike them, or you can take actual patient samples and correlate them between assays, and you can get a very good readout of whether you can measure drug. And I have no doubt about the existing assays on the market to measure infliximab can do that. The problem is the anti-drug antibodies, and we know that from the pivotal study, the Inflectra study, that the assay is nowhere for anti-drug antibodies is no way comparable to the reference assay that was used in-house in originally Centocor, ultimately J&J, and then was cross-validated by Prometheus. And to determine the cut point for clinically meaningful immunogenicity, it took a multi-center group 5 years, 1,400 samples, and we determined the cut point because you got to walk it back to PK and clinical endpoints to do that. And that has not been done with any of the biosimilars.
If I'm not, I just have a question for you.
I don't know if I would disagree with my colleague on that. So as part of the assessment of immunogenicity, besides the clinical readout, if there's any hypersensitivity reaction that occurs for a given product, the analysis of anti-drug antibody is conducted, as well as what those antibodies do. Some anti-drug antibodies do nothing in terms of their effect on the product. Some are neutralizing, and this is what's concern because this would result in decreased efficacy. And so the neutralizing antibodies are also part of the measurement.
Yeah. I'd say that the problem there is that with the drug-tolerant assay is that you can detect anti-drug antibodies in the presence of drug. And we don't know what those mean. And if there are differences in those things from the reference product, it can be a problem. And the notion that it's just all about neutralizing antibodies, I don't think is fully correct, in that if you form antigen antibody complexes to other idiotype, not just anti-idiotypic antibodies, but to other molecular configurations on the antibody, then that can result in complexes and increased drug clearance. So, it's complicated.
But so PK is measured.
Yeah, but what determines a significant difference in PK? In an individual patient? Yeah, okay. Well, I don't see that. I don't think you can translate generic drugs to biosimilars.
We can move on. We'll discuss this later.
So, David, you had a question.
I did have a question for you, Stacy, related, but and you can clarify. My reading of the interchangeable position of the FDA also said that the switching study would need to be performed in the indication of interest. Is that true? So in other words, would they have to do switching studies in Crohn's disease, or is it also going to be extrapolated?
So, the potential for extrapolation exists for a product that's seeking interchangeability for a given, biosimilar that's seeking that designation. I think, it would be, as with all biologics, whether they're standalones or biosimilar, it's case by case, so it depends on the particular product. So I can't answer that question.
It's possible that it would need to be done in the disease indication of interest.
Of interest to what? I mean, right, you want the most sensitive patient population, the most sensitive assay in which to determine the effect of the switching in terms of immunogenicity and then PK and PD if they're relevant.
So safety is a big issue, and a couple questions have come up. Is there any registry that's going to be formed or a way to track things? I know the initials and the agents and infliximab, the different things will help us track things. Is that going to be any particular concerted effort to do so we can see if there's any signal directly? Anyone? Or Stacy?
Well, Stacey, I don't think there's a
I am not
post-approval.
aware of any different surveillance.
There was
that
no
would occur.
post-approval requirement to perform a registry. So where would that likely occur? Probably among the rheumatologists. They're fairly organized as far as, I think they'll be all over this. The gastroenterology community is not going to be able to do it. We haven't been able to do the equivalent of what the rheumatologists have done with registries.
There's a question with so many biosimilars in the pipeline. Is there any guidance in helping to distinguish efficacy, which doesn't make sense? They should be similar. So I think that the concern as well comes up if you switch from one to another, a biosimilar 1 to biosimilar 2, should we use immune modulators in those patients, thinking that we might immunize them to some degree? Because then you would have had 3 different agents perhaps. Originator, let's say, they're doing well, and somehow they get switched to biosimilar 1 and then biosimilar 2.
That definitely is a possibility that will occur. And to take a step back, and I really tried to emphasize this in my talk, but the foundation for approving a biosimilar is the analytical comparative data between the biosimilar and the reference product. And so there's a slew of analytical assays that measure the heterogeneity within the reference product and the biosimilar product. So, while we, there's if you think of a chromatogram and the distribution of the molecule in terms of size and charge, the vast majority of each individual molecule will be identical to each other. But then you have variations on the tails, right, of the distribution. That goes for the reference product. It'll go for the biosimilar product. Keep in mind the reference product throughout its lifetime, once it's approved, manufacturing changes occur to that reference product, and that may make some, little differences that weren't, initially part of the approval package. Nevertheless, it's comparable. It continues to be prescribed. There's not evidence that these are causing, impacts on safety and effectiveness. It's the same with biosimilars. So in the case where you have, if it were to happen in the future where we have multiple biosimilars on the market and a patient receives a biosimilar as their initial treatment and then they are switched to a different biosimilar, it's the same premise. There's the heterogeneity should be highly similar between the biosimilar and the reference product.
The transitive property of biosimilars.
Yeah, I don't think I'd agree with that, and I'll try to walk you through it. Okay, so when you measure analytically the fucosylation patterns, you're not really measuring the difference potentially in the quaternary structure. You can't see down to the level of the individual molecule. And we know there are huge differences in the fucosylation patterns. Whether that translates into clinical immunogenicity or not, we don't know. And you know, this issue of the drift within the reference product, which is well established, no one has taken batches and then looked into all the patients exposed and seen whether there's differences in immunogenicity. No one's done that.
Like I said, there's no one looks for the reference product when it's licensed at every individual molecule because it's impossible. You know, the standard is that there are the products are highly similar in the degree to which that can be determined. And as time progresses, we're going to get even more and more advanced technology to do these kind of analyses. But right now we are at a very advanced level, and there's little question about I think fucosylation amounts are pretty easily identified, or afucosylation amounts if effector function is important. You know, there's just a lot of information that we know analytically.
Gary, let me just ask Brian a question. Brian, given what you've suggested and we have known from our own experience with just our reference agents, if a patient is switched, would you recommend that we proactively check a therapeutic drug level and look for anti-drug antibodies?
Well, in the absence of validated anti-drug antibodies specific to the agents that have been validated, your best is the PK, you know, and whether that's the point. I mean, if you know that the patient is being non-medical switched between multiple agents, then probably in an individual patient, the preservation of the PK, because I believe you can compare across assays for drug level. So it's probably your benchmark.
So maybe that would be a
There have
cautious
been
way to do
several
this.
studies done looking at different assays, at least 3 different assays. The Europeans have done so and otherwise, and they're rather similar when measured. They're not large numbers of patients, but they're clearly been published, and they're not mainstream journals like Gastroenterology or American Journal of Gastroenterology or the IBD Journal, but they've been in some European journals. So I found these studies because I had to debate someone before at another meeting related to this. Dr. Wolf?
Yeah, Doug Wolf, Atlanta. Thank you for being here, and yeah, this overall discussion is just awesome. I did want to say that you know, IBD patients are certainly different from RA patients and many of the biologics in that they are. RA patients are typically on methotrexate, and again, increasingly, especially as a result of dialogue at this meeting, this 2017 meeting, I think increasingly we're thinking about monotherapy with the anti-TNF. So I'm just thinking that it is different, and so the immunogenicity risk with these switches may be greater with IBD patients because they're not going to be as protected as RA patients because of the lack of methotrexate or an immunomodulator in many cases. So I think that one of the big concerns we have, and maybe it's real or not, is the potential for immunogenicity with switching.
Thank you, and hopefully, again, the analytical package that is submitted with the biosimilar BLA would demonstrate clearly that the product is highly similar to the reference product, so that the cause of concern for there being a different immune response after receiving the biosimilar should be diminished based on the fact that you have analytical similarity between the products.
Well, I think the fact, Stacy, that the agency has advocated for designation of interchangeability, that you have to do a proper multiple switch.
It's in the law.
But I guess the thing that I'm confused in Canada and I'm confused in the United States about the fact that the federal statute is there, or the requirement is there, and yet I hear that the states are just going to do what they want, and in Canada the provinces are just going to do what they want, despite the fact the federal government has said this isn't a good idea, guys. I don't know what to do about that.
It's happening already.
How does that happen?
Any further questions, please come up and address. And If not, we'll end the session. I'd like to thank our speakers, Dr. Rubin, Dr. Fagan, Dr. Ricci, for a terrific overview. And again, the AGA and the Crohn's Colitis Foundation for supporting this venue.